Hey everybody. So this is our first lecture. We're going to talk about virology, the history of virology, greatly simplified, but we'll hit a lot of things and just some of the basics of how viruses work, how they grow, how they infect things, to get us started on our study of virology. So let's get into it. The history of virology is really, really interesting. Humans have been dealing with viruses basically since our existence. Viruses have probably been around long before that, infecting other things. But the human history, when we actually record it, we can go back to about 160, 170 AD, so shortly after death. The first big plague that we have a history of was it's often called the Antonine Plague, but the Galen Plague gets talked about too because Galen was the one who actually recorded that, so that's what our records are based on. And it's from Roman troops were returning from the Near East. So they were in Turkey, Egypt, that Near Eastern area. And nobody knows if it was smallpox or measles. It was probably one of those two because they're so infectious. And that killed 5 million people. The next big recorded epidemic was, you're going to see a through line here of smallpox, why it was such a big deal to get rid of it. So smallpox, there was a huge epidemic in Japan. So remember too, these are ancient cultures, and so the actual total populations are a lot smaller than they are now. So to lose 5 million people was massive. The Japanese smallpox epidemic in the 700s, there's a twinge of racism to it. So they claim that it was a Japanese fisherman who was stranded in Korea, and then he brought it to the mainland. There's no real evidence of that being true, but everybody likes to be a little racist. And he brought smallpox back apparently and killed 1 million people out of a population. I believe that was like a third of their entire population or something crazy like that. Smallpox again crops back up. So now we cut to another almost, 1,000 years later? That's crazy. So we've heard all about this. So once Western Europe started trying to find the New World, they started intentionally and unintentionally using biowarfare. So as the Europeans traveled to mostly Mexico and the Caribbean, and eventually to North America as well, they spread smallpox. That was the big one. Other diseases as well, but smallpox was the worst one. It's basically responsible for the entire destruction of the Incan and Mayan empires, which is absolutely crazy. So we don't actually know what the mortality was. It's in tens of millions. So smallpox, up till now, we've eradicated it, but in the 70s. But up until now, it's probably killed hundreds of millions of people. But one of the biggest ones, it was like a mega death event, was smallpox being introduced in the New World. Now, there are other diseases too. So yellow fever is a big one. It's still here. It's been around for a really long time, for hundreds of years. I mean, it was probably around there before the 1500s as well. It was spreading more with the African slave trade, and it's a vector-borne disease, which we'll be talking about later. So when something's vector-borne, that means it's usually driven by a arthropod, like a mosquito, or like a tick, or a flea, something like that. Mosquitoes are the worst ones. Aedes aegypti spreads a lot of diseases. They're really distinguishing. It's easy to tell an Aedes aegypti mosquito because they they look almost like tiger stripes or tiger spots or something like that. Like they look really weird and intimidating and scary, as opposed to, I don't know, Aedes albopictus or something. So they spread lots of diseases though, and they hopped on the boats and came with the slave trade and spread through North America and all the islands. The islands are where there were a lot of really big outbreaks. The estimated mortality was in the hundreds of thousands. We still have tens of thousands of deaths, not just infections. There's way more infections, but tens of thousands of deaths in Africa. When I traveled to Africa this last summer, we were not in an endemic area, so we did not have to get a yellow fever vaccine. But some areas are endemic there, so that was pretty crazy. So a little more current, HIV, it's probably been spreading since the 1930s, actually. There's some debate about how it actually jumped from a simian virus. So SIV, which is simian immunodeficiency virus, still exists in monkey populations. Um, but it's related to HIV-1 and HIV-2, which spread. Uh, there's debates about how it actually spread. It's probably through bushmeat consumption. But to actually jump that species barrier, there's some debate that the oppression of the people in those areas at the time led them to, they weren't healthy in general, and then they were eating bushmeat. And it's a lot easier for viruses to jump species barriers when your immune system is somewhat compromised. Not fully immunocompromised, like if you had some kind of immunocompromised disease. Um, but if you're nutritionally deficient, if you're malnourished, then your immune system just doesn't work as well. And viruses can take advantage of that. Now, in the 1980s is when we actually isolated HIV, which I have some more stuff about history. This is just the history of pandemics right now, just to show you the gravity of what we're dealing with. But in last year, well, right now it's 2020 right now. So in 2018, 1 million people were dying every year. It used to be a lot bigger than that. Overall, we've lost 32 million people to HIV, mostly in Africa. That's crazy. That's a lot of people. There's a lot of history to that we can talk about later. Influenza A, that's where the most pandemic strains are driven, not driven, most pandemic strains of influenza are actually influenza A. There's A, B, and C. Influenza A is the one that can infect waterfowl typically and swine, so pigs, and humans. And so once we get some shifting through there, so that's where we'll have our initial viruses. If they drift, that's just basic mutations. So point mutations, small nucleotide polymorphisms, as they just, because the enzymes that replicate the RNA of influenza just aren't perfect at it. And so they make mistakes. And you get some selective pressure that drives mutations. However, when you swap entire segments, so of influenza genomes are segmented, there are 8 segments. When you start swapping segments, we call that a shift because it's so fast. And this happens when you have a superinfection. So if you have a viral superinfection in, let's say, a pig. So let's say a duck sneezes an influenza virus, the black one, onto a pig, and the pig already had an influenza virus, and they get into the same cell. The baby cells, the baby viruses, the virions that they make, can be reassorted. And that new virus is completely different. Well, it's not completely different. It's a mix of the two. It's not slowly mutating. It's a very fast mutation. And that's how we get these pandemic strains that can just spread really rapidly. We've seen a few of those recently. H1N1 comes to mind. But influenza A has been infecting people for a long time. The 1918 flu is probably the biggest one that people think of. Influenza hasn't been as big an issue until relatively recently in human history. So once we start getting that globalization, we start moving people much more rapidly than we were. And we're also having humans and animals in closer proximity and in larger quantities. And some of the practices for large-scale farming like that are not super healthy for anybody. And so when you're not as healthy, you're more susceptible to diseases. And so that's when we start getting these larger shifting events. The 1918 flu took advantage of World War I as well. So that spread, people were really sick. And there's a really good YouTube video about this that I'll post. People were really sick and then they were immunocompromised. They're fighting wars in these trenches and it's really gross. And they got sick, it started killing people, and then they went back to be rehabilitated, and then they spread it back in their home countries. And we lost 50, 60, probably more than that. Those are always underestimates. Probably closer to 100 million people from that infection. That has some really interesting correlates to the current coronavirus outbreak, in that people did a ton of quarantining early on, and then people got really sick of that after, I don't know, a few months. And then you had a second wave of infections that killed a bunch of people. Ebola it's scary because it's so transmissible. You get it on you, you're probably going to get sick. But it's pretty self-limiting. So that's the one nice thing about Ebola. It had a lot of press. What was that movie with Dustin Hoffman. It was an outbreak. That was sort of an Ebola-esque virus. It scared a lot of people. It is really scary. It can kill a lot of people, but it's pretty self-limiting because it does have high mortality, but it's very visible. Like it's really obvious and very quickly obvious that you're sick with Ebola, so we can avoid you, basically, is the advantage there. But we, up until like 2014-ish, which is pretty recently, there hadn't been a big outbreak. The largest outbreaks had been a couple people, I think, maybe 10. I'd have to look that up, but in 2014, and there have been a couple other ones. There's still current infections. There's been 10,000, so thousands of people got sick in Africa. And so that was really scary because it just kept spreading and it was hard to isolate people. There were a lot of issues with that. The medical infrastructure is not as good, but a lot of the issues were coming from people. When people would die from the virus, they were trying to take care of their loved ones' bodies, and but their loved ones' bodies were full of this super infectious virus. So that wasn't great. It transmitted a lot that way. Right now, coronavirus, there have been 3 big outbreaks of coronavirus. SARS in 2002. When I was in graduate school, MERS had just come out. I think that was 2012-ish. So that was coming around when I was taking virology courses in grad school. We used to talk about that. So SARS is almost identical to the current coronavirus outbreak that we're dealing with. And how it worked, that it jumped species barrier in a Chinese market, which happens all the time. I mean, that's basically where influenza A is getting its shifts from as well. But the current outbreak, it's pretty bad. We're supposed to be social distancing, but nobody's doing that right now. As of today, a bunch of states are opening. Some states are not. People are really upset that some of their states are not. Like my parents, they live in Michigan and they're being kind of more hardcore about it. But I would be very surprised if we don't see a big jump because the virus doesn't care about your civil liberties. It just cares about infecting people. It's been very, very successful. We will talk later about what makes a successful virus, but the coronavirus has been very, very successful. If you want to come at it from a really dark angle, it's been an awesome virus because it doesn't kill most of its hosts, unlike Ebola, and it is very infectious before showing symptoms, if you ever show symptoms at all. That's the mark of a very successful virus. Influenza does that as well. Measles does that as well. To be infectious before you show symptoms is really, really good. That's what a successful virus will do. Now, there's Ebola down there. It looks cool. Cool picture. Oh, something I meant to mention. I didn't even look at this picture. HIV, when we have our mortalities, that's really hard to estimate, because most people die from secondary infections. So tuberculosis being the biggest one. If you have HIV, it kills your T helper cells and you can't mount a good adaptive immune response. And so you end up dying from diseases that wouldn't normally kill you. So that's the history of pandemics, to give you the weight of these diseases. Those are just, that's a small list. I skipped over a bunch of viruses. And that's only in humans. We also deal with a lot of viruses in animals. I mean, coronavirus was an animal issue, at least this specific strain, and then now it's in humans. But there's a lot of interesting history to viruses. Viruses have taught us a lot about cells and biology and how things work. So I kind of pulled together some of the bigger history points. I have some better resources for the history of virology, but just to hit a few biggies. So the term virus is Latin for poison. You can dig into that, the semantics of that a little bit. It's one of those Latin words, like it means poison or a liquid that's dangerous, or like, you know, there's a few different definitions, but poison is one of them. They were using it, I couldn't figure out who was using it, but in the early 1700s, it was used to describe venereal diseases, which was probably syphilis. That was the big one. Everybody had syphilis back then. Not everybody. Syphilis was a huge problem. There was no treatment. I mean, we didn't have a treatment for syphilis until we started developing antibiotics. It was really bad. Don't Google syphilis, ancient syphilis pictures, unless you want to be grossed out, because it starts eating away at your bones and stuff. So, cut to, what is that, like 70 years later or something ish, 60, 70 years later. So, Edward Jenner developed the first vaccine against smallpox. So, he noticed that they had been doing variolation before then. Um, so, variolation is using the actual virus to vaccinate people. Not an attenuated version, but using an actual version. So that people would actually get smallpox. If you apply it to someone that way, they tended not to get as sick, but you're still giving them the actual infection. But he noticed that the cowpox virus, which is a related vaccinia virus, could also confer immunity. There was a cross-protective antibody, is what we've discovered later. One of the surface antigens of cowpox is close enough to smallpox surface antigens that the antibody against the cowpox one is able to fight the smallpox one. And so that's really nice. And so if you give someone cowpox, which doesn't really make them that sick, they're now immune to smallpox. And so he vaccinated a ton of people. There's some really weird, interesting, dark history to how those vaccines were actually administered for smallpox. Because we've had that vaccine for a really long time, even before good cell theory was around, and before good Koch postulates were actually in place. And so how we treated people, like bloodletting was still a big thing. I read this whole article about how they would take a kid, they're like, all right, we're going to vaccinate Timmy. So we're going to put him in this shelter for a couple weeks. We're going to vaccinate him. We're going to bloodlet him, and we're not going to feed him anything. We'll give them water and maybe some bread, and that's it. So somehow it was supposed to like isolate them and help them fight or develop a good vaccine response. I would argue that it was way worse to do that than actually getting smallpox. Now that might not be true. Smallpox is terrifying, but just we've come a long way, basically. So cut to almost another 100 years later, like 90 years later, Pasteur and Chamberlain. So they were in France, and France was a really strong pusher of a lot of this work. The Pasteur Institute is still one of the biggest scientific research groups on the planet. So Pasteur and Chamberlain, they were working together, Pasteur being the PI mostly for this stuff. They invented ceramic filters. Chamberlain did the work, let's be honest, but Pasteur gets a ton of extra credit for not actually doing some stuff. But you can have ceramic filters. You can still buy them. Ceramic is not the solid. It's got a lot of porous, various pore sizes. And so basically the water passes through the ceramic and then up out. And so this is how the filter would actually work. So you'd put water in, it would pass in here, and then it could drain through. That's you can study that. You can still use ceramic filters. Usually what it looks like is you would take a pot, like to a planter, a ceramic planter pot. That doesn't have a big hole in the bottom, and then you just put water in that, and then anything that drains through it is filtered. At least for bacteria, because we learned later that Ivanovsky, we'll skip these guys for a sec, Ivanovsky used a Chamberlain filter. They knew that they couldn't get bacteria to pass through a Chamberlain filter, but they could still get liquid to pass through it that would transmit tobacco mosaic disease, which we now know is caused by tobacco mosaic virus, which I have a picture of on the next slide. So they knew, oh, well, we know there's no bacteria, but it's still infectious. There was a big debate about whether or not that the liquid itself was infectious or if it was something in the liquid, because they couldn't look at it. And that was cleared up later. Just to jump back 1 year later, Pasteur and Roux were working on a rabies vaccine, and they actually developed one. And they were able to save a little kid's life. He got bit by a rabid dog, and they saved his life. Moving on a little bit, so almost 1900, the turn of the century. I'm not going to say his name right. Bayerink. He was trying to copy, as a good scientist would, Ivanovsky's work, where he was taking. They would basically grind up tobacco leaves that had tobacco mosaic disease, and then they'd run it through the Chamberlain filter and then see what the liquid did, and it still transmitted the infection. There was a lot of tobacco around at the time. Big money. And so Bayerink replicated that, and he's the first person to actually use the term virus to describe what was at the time a contagious living fluid, because they didn't know what was actually in there. So that was 1898. This date is wrong. I'm sorry. Here, hang on. The date is correct. Loeffler and Frosch At the same time, they isolated the first animal virus. So it caused, it's known as foot and mouth disease. It's also called hoof and mouth disease. This is not to be confused, as it often is, with hand, foot, and mouth disease. That's a separate virus. These guys created a heat-killed vaccine, so they were able to grow. They didn't know what they were doing at the time necessarily, but they were able to isolate a bunch of this virus. They killed it, they sort of boiled it, and then they were able to create a vaccine with it. So you can see the idea of creating a vaccine against viruses and viral diseases is very intrinsic to actual viral history. There were these really bad diseases, and we really needed to fix something. And so the idea of vaccine almost predated our understanding of how viruses actually work, which is super interesting. Let's jump ahead a little bit. So it's crazy. 1898 seems so much longer than farther distant than some of these, like 1892 to 1935, but it really wasn't. Our technology advanced so much in that time. It's absolutely crazy. So we were first able to start crystallizing things. So I tried to use people's last names just so we can get some honoring of these people. Stanley was the first one to crystallize tobacco mosaic virus. And here it is over here. This is an electron microscopy. This would be transmission, I think. Yeah, it feels like transmission, electron microscopy. Of tobacco mosaic. So tobacco mosaic virus is this really long, cylindrical, strange-looking virus. We'll learn later, we're going to talk about plant viruses versus animal viruses. And plant viruses are very strange. They tend not to have envelopes. They don't have phospholipid bilayers on their outside. And they have to be introduced usually by an insect or some kind of an event like that, because plant cell walls are so thick with that nice cellulose layer. They actually have a cell wall, unlike our cells. And so to get them through there, they need some kind of physical disruption usually. So that's why they don't have phospholipid bilayers, typically. A couple of years later, I mean, people were hardcore just going at this. So Thieler, he made an attenuated yellow fever vaccine. Basically, attenuation is when they would take the vaccine and they would passage it, is what we call that. Passaging is where you get the virus to infect a cell, like a cell line, just cells growing in a petri dish, or an egg. Chicken embryos inside eggs are very susceptible to viruses. Kind of like how even human brain cells can get infected with viruses that they wouldn't normally get infected with elsewhere, because neurons and or babies can get viruses that they wouldn't normally get infected with in specific areas, like their eyes, or just during birth they can get viruses that they wouldn't normally get. And so we're kind of taking advantage of that to passage these viruses. And so you would take this virus and put it onto a chicken embryo, and it would grow, unlike normal. Like you could just put it on a chicken, it wouldn't grow, but you could put it on this embryo and it would grow. And you could do that over and over and over again until the virus just doesn't really grow in humans very well anymore. He won the Nobel Prize like 20 years later, well, like 15-ish years later, 14. And that's great. The yellow fever vaccine was a really awesome vaccine. It was one of the first like really good, great attenuated vaccines. I put a link here to a really good article in Ethics. It's the American Medical Association Ethics Journal that talks about the history of some of this research. You may have heard of Walter Reed Medical Institute. That's the Army's research place. So Walter Reed himself created his entire career around yellow fever vaccines. The yellow fever vaccine and studying how it actually worked in extremely ethically controversial studies of American military personnel in Cuba. They just would, they needed a control group. They needed people who had the vaccine, and they needed people who didn't have the vaccine, and then they would infect them all with yellow fever. So they were infecting people. They would just take yellow fever infected mosquitoes and let them bite people that didn't have the vaccine, and they got really sick, and I think some of them died. That's not ethically okay, but I don't know, we learned a lot about vaccines, so but it's a really interesting article. You should read it. We were getting, so let's jump ahead a little bit. We're going to go a little bit faster. I'm skipping over a ton of stuff, obviously, but Hershey and Chase were one of the most important experiments that gets talked about They proved that in viruses, the infectious material was not the protein, but it was the DNA. And so we're starting to learn a little bit more using viruses as our tool about how cells and viruses and life actually works. Up until then, nobody was really sure that maybe the protein is actually what's infecting the cells and allowing it to keep on growing. Now it sounds kind of silly. We know that DNA is the infectious part. So what they did was they would label different things. So they either labeled using radioactivity at the time, which is terrifying. They're saying fluorescence intensity. It was radioactivity. And so they would take a bacteriophage, which infects, it's a virus that infects bacteria, and they would label it. So they knew that I'm going to butcher this. I think it was the protein they knew would have sulfur in it. And then the DNA they knew had phosphorus in it. And it wasn't in. So like the protein didn't have the phosphorus, and then the DNA didn't have sulfur. And so they would use radioactive phosphate and radioactive sulfur. So they knew that the DNA would have radioactive phosphate, and if it infected the bacteria, then they would look in the bacteria for what was actually in there. So they knew, like, okay, we only put in radioactive phosphate, and that was in the bacteria. The bacteria had radioactive phosphate. When we used radioactive sulfur, it stayed in, was it sulfur? I feel like it was sulfur. That stayed in the protein. So there was no radioactive sulfur inside the bacteria, but there was radioactive phosphate. And so they knew, well, there's only the DNA has the phosphate, so therefore the DNA is what's actually infectious. Really, really important study. So we jumped to the 50s, 60s, 70s. There was a lot of interesting stuff going on. Basically, as technological advances happened, then we could take those technological advances and use them on viruses. A lot of vaccine work happened in the 50s and 60s. So Jonas Salk was the big one. He had a heat-killed, or was it formaldehyde? I can't remember. But a killed polio vaccine. Polio was a huge problem. I didn't list that under our pandemics because it wasn't necessarily killing people. That's what I was focusing on for the pandemics. But it hurts people a lot. It's really, really bad. We're almost eradicated it. It's so close. It's just one of those holdout ones that's really hard to get. He had a killed one, and it took another 9 years for Sabin to actually create an oral one, which gets into vaccines and how they're administered is really, really important. Heat-killed vaccines often, they're just harder to administer. You have to inject it. You have to have somebody who knows how to use a syringe, which not everybody does, to do intramuscular injections. So if you could anybody can just drop a couple drops of sugar water in a kid's mouth. And so that polio vaccine was huge. Now there were some issues with kids having revertant strains. So this was an attenuated strain of polio. So it was still infecting you, but it was attenuated, just like that yellow fever vaccine, where it just wouldn't make you sick. But it would still infect you, which is really good because your body sees an infection and attacks it really fast. Killed vaccines, they're not as scary to your body, and so we don't have as good of immune responses against them. So you need to use adjuvants, which just basically cause inflammation. But that oral vaccine was great, but you can have revertant strains. It's pretty rare where the vaccine strain will revert back to an infectious strain or a more severe infectious strain, but it's super rare, and it was basically honestly worth it to get so many people vaccinated. So now we cut to another 10 years later, the measles, mumps, rubella. Hilleman is amazing. He is responsible, I think it's 30 vaccines he made over the course of his work at, I don't remember where he was working. Hang on. He was working at Merck. He created the first measles, mumps, rubella vaccine. Super important. I mean, that saved so many lives. Measles is terrifying and kills people like crazy. It still kills thousands of people every year. So anti-vaxxers, stop being dumb. Sanger, so a little bit more, you'll see we're dealing with the viruses themselves and how they hurt people. But then we've also got like Hershey and Chase, and then Sanger was a bacteriophage, so a virus that infects bacteria, was the first entire genome sequenced, and Sanger did that. So we still talk about Sanger sequencing. That's a way of sequencing DNA. And he did that with a virus first. So the first whole genome of any creature on the planet was a bacteriophage. Do they count as creatures? I think so. So up until now, we had been vaccinating people against smallpox like crazy, but we went hardcore in the 70s. It was one of the great triumphs of humanity to go around and eradicate smallpox. It was awful. Smallpox, I mean, let's be honest, it's killed millions for hundreds or thousands of years. So to actually get rid of it was awesome. That was a big deal. We don't have to get the vaccine anymore. It wasn't a great vaccine. The smallpox vaccine, if you talk to usually baby boomers, the only people you can see it on now, like my mom has a scar. You get a scar because it still sort of infected you a little bit. So on their shoulders, they'll have scars. So that's kind of nice. We don't have to do that anymore. Now, a little bit more info. HIV is going to come up here. So it's the 80s. We're hitting the 80s. There were probably infections passing through humans, but it wasn't until the 80s that we actually started to really notice something was going on. And that's when we started learning more about retroviruses, which HIV is one of a bunch of retroviruses. So that's where they have an RNA genome that they convert to DNA and then insert that into your DNA. So you have it forever. Human T lymphotropic virus 1, Bob Gallo, Robert Gallo, but he goes by Bob. He's still around. He was the first one to discover HTLV. There's a lot of weird, interesting history of the discovery of HIV itself. There's some controversy around who actually discovered it. Montagnier and Sanusi or whatever, they were given the Nobel Prize in 2008. So they waited like 30 years to give it to him. Or 20, whatever, which is a really long time. But it was a huge controversy that Gallo was not included. They actually included another person who I think they were working on the HPV virus that they shared the Nobel Prize with. So there was just a virus year that year. But they can only give it to 3 people. Bob Gallo should have gotten it. He got a bunch of awards. He's famous, whatever. But HIV was a big deal. Really interesting history there because, well, I think I have some more stuff to talk about with HIV. Well, we're going to spend some time talking about HIV. Basically, the reason it spread like crazy was because we were ignoring it for the first 10 years. There's some very famous stories of the Reagan era health advisors actually just laughing about it. They're like, some kind of thing that affects gay guys. Not a big deal. And then it started infecting a ton of people and it's killed millions. Probably because we just kind of dinked around, which sounds really familiar. Not taking a virus very seriously until it's too late. So late 80s, Harlow and Livingston were really important. So we knew up until now that there were some viruses or oncogenic viruses that cause cancer. That's true. We knew that before this. But Harlow and Livingston were showing that some viruses were causing cancer by actually influencing the cells themselves. Not the virus itself would influence the cell to have cancer, not directly cause the cancer, if that makes sense. And so they were influencing like tumor suppressor genes. So a virus might turn off a tumor suppressor, which allowed the cells to become cancerous. So it was kind of scary. That was a big deal, though, to show that. I just jumped another like 11 years, skipped over a bunch of stuff. West Nile virus was a big deal. It wasn't around. I mean, I've been around since, 86? So, I mean, I'd never heard of it. But late 90s, 2000s, West Nile started spreading, and it's now like everywhere. It's an interesting flavivirus, mosquito spread. As climate change happens, I don't know why people don't believe that it's happening. It definitely is, because we can track it through various different ways. Mosquito spread is one of those. So as climate, basically mosquitoes don't like it cold. So as things warm, various species of mosquitoes start moving more northward. And so we're seeing more of these diseases. Hopefully we won't see dengue in the US. I think I watched a thing about somebody getting dengue in Florida. But That would be a really scary one, but that's usually driven by the vector, not the virus itself. The virus doesn't care, but the mosquito does care. Um, 2006, I met the first guy who made this rotavirus vaccine when I took a vaccine class at Penn. So that's when we first approved the first human papillomavirus and rotavirus vaccines. That's pretty late in the game. Rotavirus still kills tons of kids. It's, I believe, the major cause of diarrhea in children under 5, which is one of the main causes of death in children under 5. Rotavirus kills tons of kids. And it's an oral vaccine, which is nice, so it's easy to administer. My kids, I think my son's going to get one tomorrow, actually. He's 6 months old. So jump ahead another almost 10 years. That's when we had that Ebola jumping out, and that was really scary. But for a virologist, super interesting. Hey, there's rotavirus over here. I didn't even talk about this. There's there's 2 viruses. There's HIV over here, an electron micrograph transmission, and there's HTLV1, which are different. Up until then, I believe we called HIV1 HTLV3 because there's HTLV1 and HTLV2. We knew it was T-cell lymphotropic, but we changed it to HIV to, that was a better name, I guess, because it was causing immunocompromising. Though HTLV can do that too, so whatever. Zika, I have an interesting story about Zika that we'll talk about later. There's a paper written by one of my professors. He got it in 2008 on a trip to Africa. And when he came home, he gave it to his wife because they did what husbands and wives do when they've been apart for months. And she got Zika virus via sexual transmission. He wrote a paper about it, and everybody laughed him out of town. Not out of town, but I was at a conference actually where they were making fun of him in 2010, because it was like 2 years before, and they were like, "Oh, sexual transmission to your wife, huh?" And you're like, "Well, I mean, of all the people to give it to, that seems okay." Not to give it to her, but like, I don't know. Well, at least it was his wife and not his mistress. And then cut to, 10 years later, and we found out that sexual transmission of Zika was a big deal, has probably been happening and causing microcephaly in children all over, especially South America. Yeah, turns out they probably shouldn't have been laughing at him. He was an interesting guy. I took epidemiology with him. What is that period there? So now, coronavirus, COVID-19, as my parents like to say, the COVID. We found it in December. It actually might have been transmitting earlier than that. China wasn't doing a great job talking about it, but we also had removed some surveillance from a lot of places. Some stuff ran out, some money was cut. There's people are playing the blame game, but honestly, it doesn't really matter now because we didn't do anything about it. A lot of countries. So we saw it in December, January, February, March. It wasn't until late March, early April that we started doing anything about it, and it was too late. Some countries dealt with it real fast. Honestly, a lot of those countries actually deal with more viral infections that spread through the populations. They deal with those kinds of things, like dengue comes to mind, or chikungunya. So they're better prepared, and their populace is better educated about viral spread. The United States, we're a more temperate climate. We honestly don't deal with a lot of those things as often. And so I think that's actually where some of this, not only pushback, but just lack of education in general about how viruses work is coming from. So that's some of the basic history highlights. I'm going to make a separate video after this one to give myself and you a little break, and we're going to talk about viruses and what they are and how they infect things.